Hello， 大家好，我是劳劳。今天呢，给大家介绍一篇来自 Berkeley、UCSF 还有 MIT 合作的生物医学领域的文章。这一篇文章呢，介绍的是怎么样去 dissect 这个癌症的进程中的事件，以及还原它们的转移史。那么癌症的进程呢，其实就类似于进化过程，从原发性到转移，再到最后发展出抗药性，其实是包括了数个事件，这些事件组成了一个非常复杂的癌症的进程。因为癌症细胞它具有一些世界上的随机性、混乱性，以及在正常的细胞中，癌症细胞其实可能比较稀缺，整个癌症进程会比较长，所以我们很难有效的还原出每一个事件，并且对它们进行观察。那么了解这些事件的分子机制以及它们的时间的发生的 timeline， 其实对于我们了解癌症的生物特性以及研发有效的抗癌手段都是很有意义的。今天我介绍的这篇文章呢，其实就是对这些事件进行了一个非常具体有效的追踪。他们利用了 CRISPR-Cas9 技术，对于原发的癌细胞群中的每一个子细胞群进行了标记，就像是一个 barcode。那么这些 barcode 呢，就是这些子细胞群的一个身份证。拥有了这些身份证之后呢，研究者就可以在这些癌症发生了转移之后，比如说从原发的位置转移到肺或者是肝或者是其他的位置之后，他们可以从这些位置中收集到癌细胞的混合物，并且对于其中的每一个细胞进行单细胞测序，他们就可以有效的建立这个子细胞群的进化树，以及还原整个癌细胞这个转移的历史。在这个视频中呢，我也添加了英文字幕，方便大家对一些新的术语进行了解和熟悉。如果大家有任何的问题的话，欢迎大家在视频的下方留言和讨论。如果我有时间的话，会尽量回复大家的每一个问题。
Hi everyone, I'm Lu. Today I will present a paper from a collaboration work done by Berkeley, UCSF, and MIT.This is an amazing project that first time combining the CRISPR-Cas9 technology and single-cell sequencing to trace the cancer metastasis and unmask the underlying mechanism of such events. And firstly, let's have a look at the background of the study. Fundamentally, cancer progression is an evolutionary process. As shown by this figure,The cancer progression occurs by a cell clone acquires some genetic change over time. For example, the activation of oncogenes or inactivation of tumor suppressor genes. After the initiation, this mutation will be inherited and more mutation might be introduced in the new generations. Simultaneously,The tumor population will be expanded by proliferation, metastasis, and reseeding of some clones back to the primary sites. Therefore, the whole process of cancer progression is highly like the evolutionary process. Metastasis is a critical step in cancer progression since it's chiefly responsible for cancer-related mortality. However, it is difficult to dissect this event, especially in real time, since it happens rare and transient and is highly stochastic. Researchers employed different methods to trace the transition of cancer cells, which can be mainly divided into retrospective and prospective approaches. The retrospective approaches check the shared sequence variations in subpopulations to infer tumor ancestry.But this method has technical limitations, such as the low resolutions and heterogeneity in bulk tumor samples. On the other hand, researchers used markers to label progenitor tumor cells and trace their changes in subpopulations. This is a good way to observe their transition. However, this method is incapable to identifying the features of subclonal tumor.To address this issue, this study first time combined CRISPR-Cas9 and single-cell sequencing to label stem cell tumor cells and dissected their subclone populations after proliferation and metastasis by single-cell sequencing technology. So the dynamics during transition and the fine details of subpopulation can be revealed. This is the schematically illustration of strategy, and I will divide this into 3 parts to introduce the establishment of lineage tracing system, the reconstruction of phylogenetic trees, and the underlying mechanisms of tumor metastasis. Firstly, the investigator used 4 rounds of lentiviral infections. Lentivirus is a general system to deliver genes into target cells, as they can integrate the genes to be expressed into the host genomes. After infections, the integrated genes will be repeatedly expressed in every generation of cells. So they first infected the adenocarcinoma cells with lentivirus carrying luciferase reporter. These integrations allow them to observe the metastasis of tumors by live imaging. So during 2 months of experiments, the distributions of tumors changed from primary injection sites to more tissues. Then they infected the cells with lentivirus carrying Cas9 protein with a M-cherry fluorescent reporter. Cas9 is the protein that can bind the target site and cut it. And the 4th step is to introduce the guiding sequence for a Cas9 protein to allow them to bind to target site. They modify the guiding sequence for one mismatch with target site to reduce affinity. To avoid the system generate too many mutations in every generation, which is the slow lineage recording kinetics, the 3rd step of infections is the most important, that they introduce target site into cells for CRISPR-Cas9 to create indels. So they modify the original CDS to introduce 3 sites every target. They also introduce int-BC as a uniquely barcode to label the target sites. So after 3 serial infections, every cell is estimated to have around 10 uniquely barcode copies of the target site. And the different combination of barcode were used to identify every single cell in the mixture population, and also as markers of progenitor cells to trace all cells derived from the single cell. So the target site in this system is used for tracing the evolution of every clones rather than tracing their progenitors. For example, after one proliferation, the parental cells will generate 2 daughter cells. Each of cell inherited the barcode label target sites and the target sites. By chances, Cas9 will bind to some target site and cut it. The left cells have a good double-strand repairment, but the right cell were introduced as indels, which is indicated by the red color. This indel will inherited by daughter cell further. So we can find all its children cells carry the same indel. In addition to it, more mutations will be introduced into target at different sites. So finally, the researchers can use the shared mutation and the novel mutations to determine which cells are precedent and which cells are later generation. After the lineage tracing system has been successfully established, the tracing system can be visualized using fluorescent imaging. If we overlap the fluorescent signals with cells, we can find most cells expressing GFP, indicating that most of the cells have the target sites. Compared to target site expression, Cas9 is expressed lower, indicating that Cas9 may not actively binding to all target sites in all cells in every generation. After they established the tracing system, they injected the engineering cells in the lung of mouse. They injected 5,000 cells in each mouse, and they also sampled the cells before implantation. According to the int-BC, they found that every 5,000 cells contain about 2,150 distinguishable clones, and they collected samples 60 days after implementation. They found the cancer cells were transited to 5 tissues plus the primary sites, which is the left lung. This is the total distribution of tumor cell number in each tissue, and they decided to focus on 100 colonies in the collecting samples that have more than 30 cells. They also found that after implantation, only about 100 clones can successfully engrafted and proliferated. They then do the single-cell sequencing to obtain their lineage datasets and also the transcriptional states. By doing this, They can obtain the pair information and know the transcriptional status underlying metastasis. Using the allele status, they applied a filtration to these 100 clones. They set the threshold for allele diversity, that is the varied mutations shown by each clone, and the percentage of alleles bearing indel mutations. The black dots are the clones passing the standard. After these filtrations, they used the lineage dataset and the corresponding tissue distributions to reconstruct the phylogenetic trees. Firstly, if the researchers check the tissue distribution of each clone, they find that each clone exhibits distinct tissue distributions, which is indicated the broad metastatic heterogeneity across the bulk tumor populations. So after that, they want to know how such diverse distribution were developed. They need to further explore the evolution of lineages. Researchers reconstructed the phylogenetic trees of each clone and matched their history of metastasis with their tissue distributions. And they used the CP003 clone as an example to show the phylogenetic tree relationships between all cells within the clonal population and summarize their history of metastatic dissemination between tissues. So firstly, we need to understand the Cassiopeia algorithm that they used to reconstruct the family tree. And this is a new algorithm that is reported by the same groups 1 year before this paper. So basically, this is a binary classification of allele information. In this color map, the row indicates cells and the column indicates every allele possibly bearing an indels or further. First, the algorithm searched for the indels that are present by most of the cells. For example, the red and blue mutations are expressed by half of cells respectively. So the cells will be divided into two groups based on these two alleles. Since they are expressed by more cells, it is more likely that the cells are parent cells so that their mutations can be passed to many daughter cells. If we further look into cells carrying red color, we find 3 or 4 cells have a yellow mutations and the other cell have a green mutations. So the algorithm classified them into 2 subgroups. Both are the daughter cells of the cells first having a red mutations. So by following such algorithms, they finally reconstruct the family tree that show the evolution of a clonal population. Let's go back to the clone CP3. This is a whole color map of lineage information of this clone and its phylogenetic tree. Every column is a subclonal carrying unique combination of indels, and every row is an allele bearing different indels. By comparing the phylogenetic distance and allele distance, they found the tree accurately models the phylogenetic relationship. If we further combine the tissue distribution with family trees, we can find closely related cells in family trees reside in different tissues, which means the ancestor cells physically transited from one to another tissue. But how it happened? This group also found that some clonal only reside in the primary site, but some clones distributed across all tissues. So this graph directly shows the tissue distribution of clones, and it's quite easy to say that the first type of clone have a slow metastatic rate. However, it is hard to distinguish between the second and the third type of clones, since both of them have a more complicated distribution. It is hard to tell how many times they transit between tissues, which is corresponding to the metastatic rate. In order to quantify the metastatic rate and identify the clones with a high and medium metastatic rate, they define the tree mat rate to review the actual transition rate of each clone. The below is the step of transition they calculate using Cassiopeia algorithm, and the above is the step they use a more parsimonious algorithm to calculate how many transitions the clone need to achieve the final distribution status. So using the tree mat rate, they successfully identify the clones with different metastatic rates, as shown in these figures. And they further look into individual cells of each clone to see whether all cells in one clone exhibit the same rate. By calculating the single-cell math rate, they find some clones are bimodal, such as the clone 7, suggesting cancers in this dataset exhibit diverse metastatic phenotypes both between and within clonal population. And we will come back to the bimodal clone later. They then pair the lineage information and the translational expression in individual cells to review the underlying mechanism of metastasis. As we mentioned before, they perform the single-cell sequencing of both target site with allele status and the gene expression profile, and they can relate the relational states of single cells to their metastatic status. So they search for genes that are highly expressed in cells with high and low math rate. The blue dots are genes that are highly expressed in low math rate cells. The number is the log of the ratio of their expressions in high and low math rate cells. The red dots corresponding to genes highly expressed in cells with high math rate. There are some genes that are found to contribute to metastasis. However, there are also some genes that are newly found to be contrary to the proposed roles in previous studies, such as the KRT17, suggesting this approach may unveil a new role of some genes. They then employ the CRISPR activator and the CRISPR inhibitor to knock up or knock down identified genes. Consistent with previous slides, after knock down the genes that are highly expressed in high math rate cells, the invasion rate of tumor cells were inhibited. In the contrast, if they knock down the genes highly expressed in the low math rate cells, the invasion of tumors were enhanced, confirming that these genes are driver or inhibitor of tumor metastasis. They then investigated whether math rate is the natural property of each clone before implantation or the obtained property after implantation. By mapping metastatic signature, they found cells exhibit heterogeneity of metastatic features before implantation, and genes that are found to highly expressed in high math rate or low math rate cells were overlapped with high or low metastatic signature before implantation. They further implant these clones into different mouse types to see whether the math rate is affected by implantation environment. They picked the clones exhibit highly similar identity in 2 mouse models and checked their tree math rate. They found that the rate is highly similar, indicating that although the metastatic phenotype of clones are diverse, but such property is inherited in vivo and determined even before implantation. So they go back to check how a clone can have 2 different math rates. As we shown in the single-cell math rate graph, they took the clone 7 displaying such phenotype and divide the subpopulation into high math rate group and low math rate groups. So these 2 groups display negative and positive association with single-cell math rate, indicating their metastatic capacity are different. They further look into the gene expression profile of the 2 modules and find 2 groups genes that differentially expressed in these 2 groups of cells. So this result suggested that metastatic rate is stably inherited. However, such metastatic rate can also evolve, displayed by the single clonal population. Finally, this group tried to use the reconstructed phylogenetic trees to mapping the tissue transition routes of cancer cells. By constructing this matrix, they found some clones not only transit from primary sites to other tissues, but other clones exhibit more complicated transition routes that they first reach the medial stylum and then transit to other tissues. So by combining this matrix, they can generate PCA that display the metastatic property of each clone. For example, the clone 19 are grouped with clones that are more likely transit to medium sternum and distribute to other tissues later. And clone 95 represent the clones that only reside in lung. After they combining all this information together, they can tell that all previously proposed metastatic seeding topology can be found in a single cancer model, such as the primary seeding, reseeding, seeding cascade, and a parallel seeding. And the metastatic topologies can influence the progression, relapse, and the treatment of cancers. For example, the cancer cell can have a seeding cascade at first, After all cancer cells are killed by drug in primary sites, they can reseeding to the primary site and lead to relapse of cancer. Taken all together, this study using high-resolution linear tracing method to show that tumor cells have a rich diversity of metastatic phenotype. The approach is essential for us to understand tumor metastasis such as rates, routes, and transcriptional drivers. Further, this approach can be applied to future studies of other aspects of cancer biology, such as evolution of drug resistance and other biological events unfolding over many cell generations.